Hey guys, this is Mr. Hewitt, John Marshall High School. In case you have Mr. Riefeld, I'm Mr. Riefeld's class. Okay, we're going to review cell cycle and mitosis. We're going to start with the cell cycle first and just kind of run through a few things and then go through each part of the steps of mitosis, which would be the M phase. So go ahead and click. You want to go ahead and click to this one right here. Okay, so I'm just going to focus right now on the cell cycle. And basically just kind of hit some of the key points and then move into just the M phase of mitosis, which would be right about here, and then work through each of the highlights there. So just starting off, the first thing that we're going to talk about is the different parts of the cell cycle. And so there are three main parts that make up interphase, and that's where the cell spends most of its life, kind of doing its normal everyday cell functions. The first part of that is called gap 1 or G1. And this is basically, the basic thing here is that the cell is just kind of, it just divided. We'll talk about the C phase where it actually divides, and it's just kind of like a little baby cell. It's all cute and cuddly, and it's starting to grow up. And so really G1 stands for growth. So that's the number one thing that you're really going to know or need to know. Next. It's going to be the S phase. The S stands for synthesis. And so if you think of the word synthesis, you would need to know that you're bringing things together. So really what you're ending up doing is you're building something. And so what are we building here? Well, the whole point of the cell cycle and eventually mitosis, we need to take one cell and we need to divide it into two cells. And so if we're going to do that, we have to take the information that's in the nucleus of our cells. We have to double it, we have to copy it. So what ends up happening in the S phase is that DNA is copied or doubled, however you want to say that. Moving on to G2. So moving on past S phase where the DNA is copied, we're going to move into G2. G2, this is kind of a key point for a cell because it's getting ready to divide, if it's going to divide. G2, basically what you're going to need to know here is cells preparing to divide. So all the little things that are inside a cell, all the little organelles, you're going to have to make enough for each cell. Last but not least, we have two sections. This arrow is going to represent two things. It's going to represent M and C. So we've got mitosis and we've got cytokinesis, and we'll talk about those individually. But big point here, within interphase, which is where the cell spends most of its time, there are basically three things that you're going to have to know about interphase. Again, G1, growth, S phase, DNA is copied, and G2 is cells preparing to divide. Okay, now what we're going to do is we're just going to move on and focus solely on mitosis. Okay, so for our first phase of mitosis, we're going to focus on, again, if you're thinking about mitosis in general and you want to try to remember this, try to use the acronym PMAT. And so the first letter, P, is going to be prophase. And I'm just going to briefly go over prophase and just talk about some key highlights. The first key highlight that you're going to need to know, the safety of the nucleus is going to kind of disappear here. So the nucleus is actually breaking down. Because the nucleus is breaking down, then the DNA has no longer got that safety net around it. And so it's got to kind of pack itself up or package itself so that it doesn't get damaged. And so what it does then is it creates chromosomes. And they're going to kind of have this X shape to it. And so the first thing, number one, is that the nucleus breaks down. Number two, the chromatin, which is relaxed DNA, starts to wind up and become a chromosome. And the last thing for prophase is that the centrioles are going to begin their journey to the poles of the cell. Centrioles move to poles. Okay? So going back to PMAT, try to leave that up. Now we're going to focus on M, which is metaphase. Okay? In the original pro kind of means before, that was like the first phase. Meta, really focus on meta meaning middle, to help you kind of remember that. So really what's going to happen here, the big highlight here, is that the chromosomes are going to line up in the middle. Okay? They're getting ready, those sister chromatids are getting ready to divide. What's happened now, so that's highlight number one, is chromosomes line up in the middle. Along with that, those centrioles are now placed at the poles of the cell. And once they're there, they start releasing these cable-like structures known as spindle fibers to attach to the chromatids and eventually pull them apart. So the whole point here is you've got, on each half of the X, you've got a chromatid that's attached by a centromere and eventually is going to be split apart. So again, this was metaphase. Now we're going to move on to anaphase, the A. Okay, so back to PMAT, focusing on letter A. Anaphase, key thing here in the word anaphase, the letter A, the chromatids are moving away from each other. And so the big thing here is, the big highlight is chromatids are split apart. So again, you have your centrioles at the poles. Now these spindle fibers are actually shorting, but you can kind of think of it as a pulling action, like the centrioles are kind of bringing their cable in, pulling those chromatids to the poles. And again, notice we still have our 4 chromosomes, but each chromatid is going to become its own chromosome because now they're going to be a part of the, we're making 2 cells here. So again, this is anaphase, chromatids split apart. Last phase. Last phase is telophase. So we're at the end of PMAT. This phase is a little bit more, there's more to know in this particular phase. And so the first thing, number one highlight, is the nucleus is going to return. You'll probably say in your book the nuclear membrane, kind of the same thing. Your chromatids now become your chromosomes, except they're going to be more relaxed. So point number 2, chromosomes turn back into chromatin. And finally, all the centrioles and all the spindle fibers, they're basically gone, they're disappearing. So spindle fibers, and centrioles disappear. And so the last thing we're going to cover today is C phase, which is cytokinesis. And so really, if you break this word in half, cyto is referring to the cell, and then kinesis is really talking about movement. Well, really what's happening here is the cells are just splitting apart. You're dividing the cytoplasm. And so what ends up happening then, your key points, your nucleus returns, it's fully formed. Your DNA is back to chromatin. And now the cell is ready to grow, and now it's going to go back into, when it's ready, G1 phase. And so the cycle has been completed. Okay? Again, highlight here is just basically the cell finally splits. Thank you very much. Have a good day. Hope you enjoyed this video.
Now copy down the following diagram and work through the steps of the cell cycle yourself.